Now these NADH and FADH2, they enter into the electron transport system, which takes place in the inner membrane of mitochondria. And this ferredoxin will also give its electron to coenzyme Q. So now see, coenzyme Q is receiving the electron from the complex 1 and from the complex 2. Hello everyone, my name is Shivansh Gaur and welcome to ATP STEM. In this video, we are going to talk about Electron Transport System. In my previous two videos, I have talked about glycolysis and Krebs cycle. If you have not seen those videos yet, I would recommend you to go and watch those videos first and then come and see this video. I will also put the link of both of those videos in the description given below, or you can also click on this I button and watch those videos now and then come back and see the video on electron transport system. So let's start with electron transport system. Now, if you have seen my previous videos, then you may remember that we have discussed that before electron transport system, glycolysis takes place and link reaction plus Krebs cycle takes place. Glycolysis is taking place in the cytoplasm and link reaction and Krebs cycle takes place in the matrix of mitochondria. In glycolysis, we got 2 ATP, 2 NADH and 2 pyruvic acid. Then these 2 pyruvic acid entered into the link reaction and Krebs cycle, where there was gain of 2 ATP, 8 NADH and 2 FADH2. So altogether, after glycolysis and link reaction plus Krebs cycle, we have net gain of 10 NADH, 4 FADH2 and 2 ATP from glycolysis and 2 ATP from the link reaction and Krebs cycle. So altogether 4 ATP. Now if you remember, I told you that majority of the ATP production in aerobic respiration takes place in electron transport system, right? Because after all of this in aerobic respiration, we are left with only 4 ATPs. And we know that we get around 38 ATPs in aerobic respiration. So the big question is, where are the rest 34 ATPs coming from? And I told you in my previous video that they are produced during electron transport system using the electrons from the NADH and FADH2. So now let's first of all understand, where does this electron transport system take place? If you see this figure here, this is mitochondria. Mitochondria is a double membrane bound structure. This is the outer membrane and then this is the inner membrane, which has infoldings called as cristae or crista, right? This is the matrix of mitochondria. Now if you remember, this is where the Krebs cycle and link reaction take place and we get NADH and FADH2. Now these NADH and FADH2, they enter into the electron transport system, which takes place in the inner membrane of mitochondria. Okay, in the inner membrane of mitochondria, we have different complexes. Basically 4 different complexes are there: complex 1, complex 2, complex 3 and complex 4. The electrons given by NADH and FADH2, they move through these complexes, and when the electrons are moving through these complexes, the energy is utilized to pump the hydrogen ions from the matrix of mitochondria into this intermembrane space. So what happens is, as the electrons given by NADH and FADH2 move through these complexes, the hydrogen ions are pumped into the intermembrane space and they keep getting collected here. So over a period of time, hydrogen ion concentration in the intermembrane space starts increasing, which creates a proton gradient, also called as hydrogen ion gradient, where there are more hydrogen ions in the intermembrane space and less hydrogen ions in the matrix of mitochondria. And as we know that if hydrogen ions are here in higher concentration and in matrix they are in lower concentration, they would want to go from the region of their higher concentration to the region of lower concentration. right? And we will utilize this concentration gradient to produce energy, because these highly concentrated hydrogen ions, when they move through this particular ATP synthase, which is also present in the inner membrane of mitochondria, this is called as ATP synthase. So here ATP synthase utilizes the breaking down of this concentration gradient, right, as an energy source to add one inorganic phosphate to ADP, and we ultimately get ATP. So this is what is happening here, right? We are utilizing the electrons from FADH2 and NADH. When they move through this, they create high hydrogen ion concentration in the intermembrane space. When these same hydrogen ions moved along their concentration gradient through ATP synthase, it produces ATP. And that's how we get the majority of ATP production. Now the question is, what are these complexes? Complex 1, complex 2, complex 3, complex 4. What is their name and how exactly this entire process of movement of electron takes place? Okay, so see here. The complex 1 here is called as NADH dehydrogenase. Why it's called as NADH dehydrogenase, I'll come to that. But for now, I'm just telling you the names of all the 4 complexes. Complex 1 is NADH dehydrogenase. That is this one is NADH dehydrogenase. Complex 2 is called as succinate dehydrogenase. It is also linked with the Krebs cycle. How exactly? We will talk about that. So this complex 2 here that you see is succinate dehydrogenase. Then complex 3 is cytochrome C reductase. Right? Cytochrome C reductase. This complex 3 that you see here is cytochrome C reductase. And the complex 4 here is cytochrome C oxidase. That is this complex, 4th complex that you see here is cytochrome C oxidase. Okay, now we need to understand how exactly these hydrogen ions are pumped out when the electrons move through these 4 complexes. So let's start with that. See here. I'll start with the complex 1. Please remember the names, because names are very important here. This is our complex 1. Okay, this is outer membrane of mitochondria. This is inner membrane on which these complexes are located, right? Okay, and the electron is given by NADH, right? So NADH gives its electron, and when it gives its electron, it gets what? Oxidized, right? It gives the electron and becomes what? Oxidized. So when it gets oxidized, this hydrogen is also lost from NADH and it becomes NAD plus again. Fine. So since hydrogen is also getting lost here and NADH is getting oxidized, this complex 1 is called as NADH dehydrogenase, because it removes the hydrogen and electron from what? From NADH. And these electron from NADH, it goes to a complex present in the complex 1, right? This is called as FMN. In short, the full name is flavin mononucleotide. So who accepts the electron from NADH? FMN, right? So FMN will accept these electrons, right? And these electrons will be given to the other electron carriers which are present in the complex 1. So these ferredoxins which are present as electron carriers in the different complexes, basically it happens so because iron has 2 oxidation states, Fe2 plus and Fe3 plus, that is ferrous and ferric, and it is interchangeable, like it can interchange between these 2 oxidation states by giving electrons and taking up electrons. So it becomes an ideal atom or an element to carry the electrons, because it takes the electrons and gives away the electrons as well because of its 2 stable oxidation states. Okay, so see here, these FES are nothing but ferredoxins which are electron carriers. Okay, so this electron from NADH will be taken up by FMN, flavin mononucleotide, and then it will travel through the different electron carriers in the complex 1, right? And during all this journey of electron, the hydrogen ions from the matrix of mitochondria will be transported into the intermembrane space. You can see that hydrogen ions are being pumped outside. Now this electron from here, from the complex 1, will go to coenzyme Q, which is closely related to complex 2, right? It is present here, not inside the complex 2 but outside the complex 2. Coenzyme Q is present, right? This coenzyme Q, it takes the electron from here. Now see, complex 2 is very important here. See what is happening here. One electron is coming from here, from complex 1. One more electron is coming from where? From this reaction. If you remember, it took place in Krebs cycle where FADH2 was getting produced, and I told you at that time that this reaction is linked to the complex 2 of the electron transport system. And see here how it is linked. So this complex 2 basically helps in succinate getting converted to malate. Okay, so succinate gets oxidized to malate and we get FADH2. That is, FAD plus gets converted into FADH2, right? Therefore, the name of this complex is what? Succinate dehydrogenase, because it removes the hydrogen from the succinate and adds it to FAD plus to form FADH2. Okay, now this FADH2, which is electron rich, it will give its electron to again electron carriers, F ferredoxins, and this ferredoxin will also give its electron to coenzyme Q. So now see, coenzyme Q is receiving the electron from the complex 1 and from the complex 2, right? So it takes up these two electrons. Look at this reaction. Q, that is ubiquinone. What is Q here? Ubiquinone, right? Ubiquinone, it takes up two electrons, one from the complex 1, one from the complex 2, and it also takes two hydrogen ions from the matrix. Please remember that. Where does it take hydrogen ions from? From the matrix. So it takes two electrons and two hydrogen ions from the matrix and it gets converted into QH2, which is ubiquinol. Now see, ubiquinol got two electrons and two hydrogen ions, right? It got one electron from complex 1 and one from complex 2. Now this coenzyme Q, that is ubiquinol, will give its two electrons to the cytochrome C present in the complex 3. Okay, so the electrons, these two electrons, we can write it like this here, that these two electrons, right, they go to what? Cytochrome C. Then they go from cytochrome C to ferredoxin, from ferredoxin to cytochrome B, right? So after all this journey, the hydrogen ions are also pumped. During this journey, the hydrogen ions are also being pumped into the intermembrane space, and the electrons ultimately are taken up by cytochrome C, which is present in between these two complexes, complex 3 and complex 4. Now remember, what was the name of complex 3 here? Cytochrome C reductase. Why it's called as cytochrome C reductase? Because this complex 3, when it gives the two electrons to cytochrome C, which is present here, the cytochrome C gets reduced. So therefore the name cytochrome C reductase, because it reduces cytochrome C present here. Now this cytochrome C, which has received the two electrons, it will give these two electrons to the other electron carriers present in the complex 4. Now see, when the complex 4 will take up two electrons, that is electrons from the cytochrome C, now cytochrome C will get what? Oxidized. So see, complex 3 was reducing cytochrome C. So therefore complex 3 was called as cytochrome C reductase. But when the cytochrome gives its electron to complex 4, the cytochrome C gets what? Oxidized. So therefore complex 4 is called as cytochrome C oxidase. Ultimately these electrons go from copper to cytochrome A to again copper electron carriers. Then it goes to cytochrome A3, and ultimately what is happening to these electrons? These electrons are ultimately accepted, taken up by oxygen present here. So oxygen is the terminal electron acceptor. This is the role of oxygen in aerobic respiration. This is what it does, right? That it accepts the terminal electrons. Okay, and so it takes up these electrons ultimately, which started its journey. Remember, from here NADH gave the electron, FADH2 gave the electron, 2 electrons, then ultimately here, and ultimately taken up by what? Oxygen. Oxygen therefore is called as what? Terminal electron acceptor. And when the electrons are accepted by oxygen along with hydrogen ions, it forms the molecule of water. Right? Now see, you must be wondering, sir, why all this is happening? See, all this happened because hydrogen ions concentration was to be increased in the intermembrane space. So when these electrons were going through this entire process, hydrogen ions meanwhile were getting what? Pumped into the intermembrane space. Now these hydrogen ions are kind of stuck there. Why? Because they cannot move through any part of this membrane, because inner membrane of mitochondria is impermeable to hydrogen ions. It does not allow these hydrogen ions to go back into the matrix, which means they keep getting collected, and over a period of time hydrogen ion concentration increases here and it decreases in the matrix. Now these hydrogen ions badly want to go back into the matrix, because that's natural nature of any molecule, that it moves or it tries to move from the region of its higher concentration to the region of its lower concentration. And here, these hydrogen ions which are desperately wanting to go back, right? Who provides a gateway to them? It is ATP synthase. ATP synthase is like, okay guys, you want to go inside? Okay, I'm there, right? Okay, so what does it do? This ATP synthase, it provides the channel, or it provides the gateway so that these hydrogen ions can move. So when they get this gate, when they get this channel, they quickly rush through it. They move through it, and that energy, when they are moving, when they are rushing through it, that energy is utilized by this ATP synthase enzyme to produce what? ATP. That is, this energy, this proton gradient energy, is converted into the energy stored in the form of what? ATP. Right? This ATP synthase has F0 and F1 particles, right? And here, when the hydrogen ions move through this, then F1 has conformational changes, right? This conformational changes, they have enough energy to add the inorganic phosphate to ADP and form what? ATP. So this is how the NADH and FADH2 can result in ATP production by the electrons which they gave to these complexes. And that is how we get the rest of the 34 ATPs. Now see, one NADH can result in the production of 3 ATP molecules, and one FADH2 can result in the production of 2 ATP molecules. Okay, please remember this. How much ATP are produced by one NADH? 3. How many by FADH2? 2. Now the question is, how many NADH and FADH2 we had at the beginning of ETS? So see here, I told you at the very beginning only that we had 10 NADH and we had 2 FADH2. So if one NADH produces what? 3 ATP, then how many will 10 NADH produce? 10 NADH will produce, we have to multiply it by 3, we will get what? 30 ATP. Okay, then if one FADH2 produces 2 ATP, then how many ATP would be produced by 2 FADH2? we have to just multiply it by 2. So 2 ATP. How many ATP we will get? 4 ATP. We got the 34 ATPs that we were looking for, right? So we will get 34 ATPs from the electron transport system. Plus, remember, 4 ATP we already had from what? Glycolysis and the Krebs cycle. So we will add these 4 ATP as well. And total in aerobic respiration, we will get 38 ATP. Right? So we got all the 38 ATPs from what? From one molecule of glucose. Fine. So I hope this electron transport system was clear. How electrons move and how they result in energy production, that is ATP production. I hope that is also clear. With this, we have completed aerobic respiration. Okay, you save this playlist and you can go through this playlist whenever you are wanting to revise this chapter just before your exams. We'll meet you in another video with another very interesting topic. Till then, please subscribe to ATP STEM and we'll meet you in the next video. Till then, take care. Bye-bye.